Hey students, so today we are going to discuss the topic of scientific practices and tools. So, what is scientific method basically? Think of the scientific method as a step-by-step recipe for figuring things out. Okay? It is a reliable, repeatable way for scientists to look at the world, test ideas, and find out the truth. Okay? So, let's discuss its definition. The scientific method is a systematic approach used to investigate natural phenomena, acquire new knowledge, or correct and integrate previous knowledge. It's designed to be objective and repeatable, allowing researchers to test hypotheses and build a reliable understanding of the world. In short, the scientific method is like a recipe for figuring things out. So, as I have told you that think of the scientific method as a step-by-step recipe for figuring things out. Like it is a reliable, repeatable way for scientists to look at the world, test ideas, and find out the truth. Okay? So, steps involved in scientific method. Okay. Here's a case study of malaria. Okay. Earlier, malaria was thought to be the cause of bad air instead of mosquitoes. So this was a problem scientists were facing at the time of malaria outbreak. A long time ago, people wrongly believed that malaria was caused by bad air, not mosquitoes. Okay? So scientists used the scientific method to find the real truth. So the first one is observation. Notice something interesting or identify a problem. This step involves gathering data and information through your senses or existing research. So basically, what it means that paying close attention to something or noticing a problem using your senses or research. Okay? For example, one scientist observed the blood of malaria patient under a microscope and found some tiny creatures and named them plasmodium. Therefore, observation was made that plasmodium is present in the blood of malaria patient. So basically, we discussed the example of malaria. Like a scientist looked at the blood of a malaria patient under a microscope and noticed tiny living creatures inside it. And they named these creatures as plasmodium. And they named this creature as plasmodium. So the result is that the observation was made that plasmodium is found in the blood of malaria patient. Okay, so the next one is question. So formulate a specific question or identify a problem that you want to address based on your observations. So basically, what it means that asking a specific question based on what you just observed. Like, for example, so the question arises, if the malaria is caused by bad air, does healthy person has plasmodium in their blood? Like they have said that if people thought bad air caused malaria, but scientists kept finding these tiny creatures in sick people, a question naturally popped up. Like if malaria is caused by bad air, does a healthy person also have plasmodium in their blood? This is basically a main question. So the next one is hypothesis. Okay. So hypothesis or developing a hypothesis. So, propose a tentative explanation or prediction that can be tested. A hypothesis is often stated in a way that is both measurable and falsifiable. It is like an intelligent guess that is made before experiment. So, the next one is hypothesis. Okay. Propose a tentative explanation or prediction that can be tested. A hypothesis is often stated in a way that is both measurable and falsifiable. It is like an intelligent guess that is made before experimentation. So, basically, what it means that coming up with a smart, testable prediction to answer your question before you actually run any experiment. Okay, for example, in this case, the hypothesis made upon observation is that the cause of malaria is not bad air but plasmodium. Therefore, stating that if plasmodium is a cause, then all malaria patients should have in their blood. Okay. So the next one is experiment or testing the hypothesis. Okay. So design and conduct experiment to test the hypothesis. This involves setting up controlled conditions to isolate variables and gather data. There are two groups when performing experimentation, like a control group, which is healthy or unchanged, and an experimental group, which is changed or affected. So, basically, what it means that designing and running a fair test to see if your guess, which is you can say hypothesis, is right or wrong. Okay, to make it fair, science experiments usually use two groups. Okay, which is control group and experimental group. Control group is basically the standard normal group used for comparison, nothing is changed here. Okay, whereas in experimental group, the group that actually has the condition or change you want to test. Okay, like in the malaria example, scientists gathered two groups of 50 people each, like control group, which is 50 healthy people, and experimental group, like in which 50 malaria patients are involved. Okay, so they took blood samples from everyone to compare them fairly. Okay, so the next one is analysis. Examine the data collected from the experiments, look for patterns, correlations, or any anomalies that can help support or refute the hypothesis. So, basically, carefully studying the data and information you collected from the experiment to see what it proves. It is basically analysis. Okay, so here we have example. Blood samples from both groups were studied under microscope and showed that all malaria patients had plasmodium while healthy people's blood did not. Okay, so basically, scientists looked at all 100 blood samples under a microscope to see who had the tiny plasmodium creatures and who didn't. Okay, so the result is that they found that all 50 malaria patients had plasmodium in their blood while the healthy people did not. So the next one is conclusion. Based on the analysis, determine whether the hypothesis is supported or not. If the data supports the hypothesis, it might be accepted or refined. If not, it might be rejected or revised. Like you can say that looking at the results of your analysis and making a final judgment, like you decide if your initial guess was correct or wrong, which is hypothesis, and if it was correct, it is accepted. If not, it is rejected. For example, the conclusion drawn from this case is that plasmodium is a cause of malaria because plasmodium was found in every single malaria patient and none of the healthy people. Scientists officially decided on the truth, which is the conclusion is that the plasmodium is a cause of malaria. Okay, so the next one is communication. Share your findings with others. This can be through research papers, presentations, or other forms of dissemination. Peer review and replication by other researchers are important aspects of this step. For example, the results got from this experimentation were communicated to all the scientists and people in the world so that people can take necessary measures to prevent getting infected and develop a possible cure. Okay, so basically, scientists shared these facts globally so that everyone knew bad air was not the enemy. Like this allowed people to take the right safety measures like avoiding mosquitoes and start working on a proper medicine or cure. Okay? So the next one is revising the hypothesis, like back to the drawing board. So this step is taken when the experiment failed to support your hypothesis. So, you have to go to the step of developing a hypothesis again based on the data previously achieved that was failed. So basically, the summary of the whole process is that science is a loop. You observe, you guess, you test, and you share. If you are right, you share it with the world. If you are wrong, you change your guess and try again. Okay?